Hi dear students, a warm good morning to all of you and welcome to the second session of Bacterial Nutrient Uptake. In the previous session, we had discussed about the importance of micronutrients as well as macronutrients which are essential for the bacterial growth as well as various growth factors which are to be provided by the environment. Now, all these nutrients which have been required by the bacteria can be taken up by different mechanisms. We mentioned about the passive diffusion technique as well as the facilitated diffusion technique, which does not require energy in the previous session. The only difference between these two was that in facilitated diffusion, certain proteins are being involved, whereas in passive, protein involvement does not happen. Now let's talk about the next one called the active transport. What is active transport? When you hear the term active itself, do remember that they require some amount of energy for the active transport. The active transport, it resembles the facilitated diffusion in a case that it involves proteins and they bind particular solutes with very much specificity. And these carrier proteins are also characterized by the carrier saturation effect at high solute concentrations, as we had noticed that when the solute concentrations increase, the carrier molecules might get saturated. Such effect is also been present in active transport. But the active transport differs from the previously discussed facilitated diffusion in some aspects. And the most important among them is that they use metabolic energy and they can also concentrate substances across the plasma membrane. These are the two different aspects in which the active transport has been different from the facilitated diffusion. And you can see that metabolic inhibitors that block the energy production will also inhibit the active transport but do not immediately affect the facilitated diffusion. Now let's discuss the types of active transport. There are mainly three types of active transport: the primary active transport, the secondary active transport, and the group translocation. They mainly differ in the terms of energy used to drive transport and to whether or not the transport molecule is modified as it enters. So let's go into the details of it. The primary active transport is mainly mediated by carriers called the primary active transporters. And they use energy provided by the ATP hydrolysis to move substances against a concentration gradient without modifying them. And usually the primary active transporters, they have been considered to be uniporters. That is, they will move a single molecule across the membrane. So the key features of the primary active transport is that they require energy. And when the substances are being transported across a plasma membrane, what happens? ATP hydrolysis does happen, and do notice that the substance which is getting transport will not be modified when it is being transported across the membrane. That is the case of primary active transport. And the most important among the primary active transporters, as we all know, in active transport, proteins are being involved. These mostly they include the ABC transporters. Now just have a look at this. Consider this case. In this case, you can see that only one substance is being transferred and it happens only in one direction. That is, it is a uniporter. Okay, when only one molecule is being transferred across the membrane, it is called a uniporter. And in the case of primary active transport, the proteins which have been involved in the primary active transport are being found to be uniporters. That is, a single molecule will move across the membrane. And we can see that this ABC transporters are the best examples of the what? Examples of the primary transporters. Now, what is ABC transporter? It is a ATP binding cassette transporter. That is the full form of ABC transporters. Now, how do they do this? You need not memorize this figure. I just put it for your understanding. They have different domains. Okay, there are two hydrophobic membrane spanning domains. Now, if this is the membrane, you can see this. This is a membrane. They have got two hydrophobic membrane spanning domains, and also um, there are also ATP binding domains which has been found towards the what? Cytoplasm surface. This is the cytoplasm. So there are some dot, like you can see the spherical ones, which can bind to the what? ATP. ATP is being depicted in orange color over here. So when these two domains, okay, interact with the when the with the ATP molecules, and when they hydrolyze it, some particles will be brought inside into the cytoplasm. So the membrane spanning domains, they form a pore in the membrane. So you can see they are forming a pore here, and the ATP binding domains will bind and hydrolyze ATP to drive the uptake. And you can see the ABC transport employ substrate binding proteins. Now, sometimes see there are certain binding proteins also being here. These are the binding proteins. The substrate binding proteins bind the molecule to be transported and then interact with the transporter proteins to move the molecule into the cell. And E. coli, the most commonly studied bacteria, it transports a varieties of sugars and amino acids by this type of mechanisms. So you can see this is a ABC transporter with a hydrophobic ends transversing through the membrane, and they also have a ATP binding domains. So the substance which has to be transported will be brought about by the binding protein and it will reach up to the what? The ABC transporter, and this binding protein will transfer it, and what happens? Uh, the protein will come and interact with the ABC transporter, and at the same time it will interact with ATP, and the breakdown of ATP will drive the ABC transporter to undergo certain conformational changes, and the substrate will be brought about into the inside of the particular cell. This is how a ABC transporter or a primary active transporter, for example, is working. Now, next we're going to talk about the secondary active transport. In the case of secondary active transport also, what happens? We require energy. But the difference is that there would also be a potential, that is, there would be an ion gradient for the transport of the substances without modifying them. The substance is not getting modified, but there would be a ion gradient when the substance is also being transported. In other sense, they are being considered to be as cotransporters. That is, they move two substances simultaneously. That is, when the ion whose gradient powers the transport and the substance being moved across a particular membrane. When the ion and the other substance both move in the same direction, it is called the sympod, and when they move in the other direction, it is called the antipod. Now you can have a better understanding by this. In the case of co-transporters or in the secondary active transport, I told you energy is required, and at the same time there would also be a ion gradient. That is, along with the substance that has to be transferred, another substance is also being transported, and they are hence called the co-transporters. And the other substance which is getting transported is called, it would be most probably the ion gradient. And if the ion gradient and the substance to be transferred is moving in the same direction, it is called a symporter. And if they are moving in the opposite directions, it is called the antiporter. So hope you understood what is a secondary active transport. And the most well-studied example of the secondary active transport is the lactose permease of E. coli, which is transporting mainly what? Protons. Okay, the proton is also being transported, and also the sugar called the lactose is being transferred into the cytoplasm of the E. coli with the help of the enzyme lactose permease. And for this, the protein which has been involved was lactose permease, and it interacts with two different components, that is a proton gradient as well as the lactose, and as the proton gradient is been moving down, what energy is released, and it also drives the solute inside the into the cell. So that's about the secondary active transport. Now the next one which we have is a group translocation system. Do remember, this is also a type of active transport, so we require energy over here. And the mechanism in this system, it mainly involves the transport of the compound into the cell by first allowing the compound to bind with a protein on the cell surface, following by altering its chemical structure during its passage through the across the membrane. So what happens is the substance which has to be transported here is being binding to the plasma membrane, and it will undergo some modification, and then it moves across the membrane. And the most well-studied example of this is the phosphoenol transferase system, which is a distinct method used by bacteria for sugar uptake, where the source of energy is phosphoenol pyruvate. So let's take a look into this. Now here, in the case of group translocation, do remember the substance which is getting transferred gets modified as it is brought into the cell. Now here, phosphoenol pyruvate is the substance that has to be broken down. So that is combining with another protein called the E1, and it gets converted into pyruvate. And this pyruvate will be converted at the same time. HPR is being formed. Okay, so the phosphate of pyruvate is transferred to E1, and then to HPR, and then HPR to form H2A, and this H2A, the phosphate from is being transferred to H2A, and with the help of that, what happens, the mannitol is being transferred. So mannitol is converted into mannitol phosphate. The substance which is being transferred over here is mannitol. Mannitol is converted into mannitol 1 phosphate when it is being brought into the cell. But at the same time, you can also see glucose is also being transferred into the cell with the help of glucose 6 phosphate, by converting it into glucose 6 phosphate. So the substance which is getting transferred over here is getting modified as it is getting moved into the cell. The next type of transfer that we have is the case of siderophores. Siderophores are the main carrier proteins which are being involved in the transport of iron in the case of organisms. And you can see that microorganisms, they use cytochromes and various enzymes for the transport of iron. If there is a deficiency of iron in the environment which they are growing, or when iron is not being available, the bacteria will produce certain siderophores, or that they are called the iron bearers. And these siderophores, they are low molecular weight organic molecules which will bind to the ferric ion and supply it to the cell. So when there is a scarcity of iron, the microorganisms will produce siderophores. And once the iron siderophore complex has reached the cell surface, it binds to the siderophore receptor protein. Then either the ion is released to enter the cell directly, or the whole iron siderophore complex is transported inside by an ABC transporter. So you can just have a look like this. If this is a bacteria, the bacteria has receptors for the what? For the siderophores. The siderophores have a very high ability to bind with iron. So when the iron is not sufficient for the bacteria, it will produce some siderophores. And these siderophores will go and find out where is iron and bind to the what? Iron. And after that, the bacteria will either using its receptors take the siderophore ion complex. Okay, either the ion is directly taken, or the siderophore ion complex together is taken with the help of certain other ABC transports. So this is the case of siderophore mediated ion transport. With this, we come to an end of the modes of bacterial nutrition. Thank you.